Hello, I'm Dr. Stevne Pipe from the University of Michigan, and at this year's ISTH, I had the opportunity to present an update on the safety and efficacy from the HOPE-B trial. This is an investigational gene therapy for hemophilia B using Etranacogene Dezaparvovec, or Etranades for short. And this comprises an adeno-associated virus serotype 5 containing a codon-optimized, highly active Padua variant human factor IX transgene and includes a liver-specific promoter. HOPE-B is an ongoing phase 3 trial. It has dosed 54 patients and has previously reported that mean factor IX activity increased to near normal levels and abolished the majority of bleeding events over the first 6 months after dosing. So the current report is safety and efficacy now over a full year of follow-up. Most notable for this trial is that patients with neutralizing antibodies against AAV5 were eligible in this trial. And this differs from most all prior AAV gene therapy trials where neutralizing antibodies against AAV have been an exclusion criteria and resulted in a quarter to a half of trial candidates screening out for participation. 2 prior clinical trials data with this vector contributed to informing the HOPE-B study that pre-existing neutralizing antibodies need not lead to exclusion. This trial also included a lead-in phase before gene therapy dosing, where the subjects were monitored for factor IX utilization and bleeding events for 6 months while on continuous prophylaxis with factor IX. Overall, 43% of the participants had neutralizing antibodies directed against the AAV5 capsid. But following Etranades treatment, factor IX activity increased to a mean of 39% at week 26 and 41.5% by week 52. Notably, no clinically meaningful relationship was seen between the pre-existing neutralizing antibodies and individual subject factor IX levels from month 6 to 12. And with this level of mean expression, perhaps not surprisingly, 96% of subjects discontinued prophylaxis and remain off prophylaxis. The majority of subjects did not report any bleeding events during the 52 weeks following dosing. Spontaneous bleeds were uncommon and continued to decrease over time. And there was a 96% reduction in factor IX infusions. The most common treatment-related adverse event was an ALT increase that was observed in 9 of the 54 participants. This did trigger a short course of oral corticosteroids, but all subjects were able to be weaned off of the steroids by week 26 post-dosing. The primary endpoint for this trial is the annualized bleeding rate observed from 6 months to 18 months post-dosing, then compared to that obtained from the lead-in phase while they were on factor IX prophylaxis. So we're looking forward to this next data cut and its potential to lead to regulatory submission in the coming months.